Hello, my name is Mel Catalan and today I will introduce nuclear chemistry. First of all, we have to discuss what nuclear chemistry is. And it basically is a field in chemistry that involves studying the nucleus of elements and changes to it. This study can be used for nuclear reactions, which is basically a process in which two nuclear particles collide and produce new nucleides. And before I go into detail, we have to understand the atom structure. As you can see, an atom is composed of a nucleus containing protons and neutrons surrounded by a cloud of electrons that orbit the nucleus in various levels. And by levels, I mean orbitals. And these orbitals are paths that represent where the electrons are likely to be found. Now I will talk about elements and isotopes. An element is a substance that is composed of only one type of atom. And elements can have variations, and these variations are called isotopes. And the simple definition is that isotopes are a variant of a chemical element in which the atom has the same number of protons but has a different number of neutrons, which results in having a different atomic mass. But it still maintains the same chemical properties due to having the same number of protons. And to identify such variations, we use the isotopic notation, which is a way of writing the atom symbol, and it is very useful for identifying the specific isotope of an element. The way it works is that the mass number, which is the total number of protons and neutrons, is written as a superscript to the left of the element symbol, while the atomic number, which is the number of protons, is written as a subscript, which is also to the left of the element symbol. For example, I will show you the isotopes of lithium. And as you can see, the top number tells you the total number of protons and neutrons, while the bottom number tells you the number of protons. And you can see that the number of protons does not change for any of them. Now let's discuss radiation. In nuclear chemistry, radiation is produced as a result of a nuclear reaction. The way it works is that when a reaction occurs, energy is released in the form of particles or electromagnetic waves emitted from a nuclei of unstable atoms when they undergo radioactive decay. I will elaborate on radioactive decay later on, but as of right now, I will explain the three main different types of radiation. I will start with alpha particles, which are a positive large particle consisting of two protons and two neutrons, which are bound together and are emitted during the radioactive decay of heavy elements like uranium or radium, and because of their large size, they use up their energy over short distances and they are unable to travel far from the atom, and this is why they have low penetration power and they can be stopped easily. And by penetration power, I refer to the ability of radiation to pass through a substance. In contrast, beta particles are small moving particles that can be either positive or negative. They also have more penetration power than alpha particles and travel farther than alpha particles, making it possible for them to penetrate the skin and cause damage such as skin burns. Lastly, we have gamma rays, which are weightless packets of energy, and unlike alpha and beta particles that both have energy and mass, gamma rays are pure energy, and they are similar to visible light, but they have much higher energy, and they also have higher penetration power than alpha and beta particles. This image shows you the penetration power of certain radiations, and you can see how gamma rays has the highest penetration power compared to alpha and beta particles. Now I will talk about radiation decay. I previously mentioned that radiation is produced when unstable atoms undergo radioactive decay. And by radioactive decay, I refer to the emission of energy in the form of ionizing radiation, which is the amount of energy required to remove an electron from an atom or molecule. First, we have alpha decay. And alpha decay is when an unstable atom nucleus emits an alpha particle and transforms into a different, lighter atom nucleus with a reduced atomic number by 2, and this is done for the nucleus to become more stable. The next one is beta decay, and again is when an unstable atomic nucleus transforms by emitting a beta particle, which can be either an electron, which will be the beta minus decay, or a positron, which will be the beta plus decay. And it essentially works by converting a neutron into a proton and vice versa, and this process changes the atomic number of the nucleus without significantly altering the mass number. Lastly, we have gamma decay, which is basically the emission of very high energy in order to stabilize the unstable nucleus. Unlike the other types of decay, it does not involve a change in the atomic number, and it just goes from an excited state to a ground state, which is the lowest allowed energy state of an atom or molecule. Next, I will discuss half-life. In nuclear chemistry, half-life refers to the amount it takes for exactly half of the atoms in a radioactive state to decay into another element. And each radioactive isotope has its own specific half-life, and it cannot be changed by external factors. Half-life is important because it helps predict how long it will take for a given amount of radioactive substance to decline at a certain level. In order to find half-life, you have different formulas to help you for very different problems. And as of right now, I will show you an example of using the first order formula. The example says the decay constant of a radioactive isotope is 0.01 per year. What is the half-life? And you have to use the first order formula, in which you basically divide 0.693 by the decay constant, which is a given number, which is 0.01, and it gives you a total of 69.3 years. Now that we know the basics of nuclear chemistry, let's discuss a real-world application of nuclear chemistry. In this case, I will be discussing induced mutagenesis using gamma rays. Induced mutagenesis is a technique in which plants are exposed to gamma radiation in order to create genetic mutations in their DNA. This is done with the purpose of leading to the development of new traits to enhance crops, make them disease resistant, and create genetic diversity for breeding purposes. The way this technique works is that the plants or seeds are influenced by various factors when working with this technique, such as the type of radiation, which in this case will be gamma radiation, as well as the dose rate, the absorb rate, and the sensitivity of the organism. Once the plants or the seeds are exposed to gamma radiation, there can be a direct or indirect damage in the DNA that makes it possible to create genetic diversity. And once a plant or seed is exposed to the gamma rays, the plants are grown to observe any trait differences, and the process is repeated until there is an optimal trait. And by direct damage, I refer to the damage that is done directly to the nuclear DNA. The energy from radiation can create unstable molecules that break the DNA strands, leading to serious damage like single-strand breaks or double-strand breaks. And this type of damage can either be successful for variation, or it can interfere with the process like gene expression or cell division. The other type was indirect damage, and it is when the radiation splits water molecules into reactive particles called reactive oxygen species. And these molecules cause oxidative stress, leading to DNA damage, protein breakdown, and damage to cell membranes. Another type of damage is when DNA and protein create bonds called crosslinks, and this prevents DNA from replicating, which essentially prevents certain traits to be passed down. And because it is easy to create damage that can essentially either kill the plant or prevent a trait to be passed down, each lab requires plenty of research before it can have a successful result. Also, The primary source of gamma radiation is cobalt-60 and cesium-137 isotopes, as they provide a stable and long-lasting source of gamma radiation due to their half-lives. If you're curious to see how a plant will look like before and after the treatment, I will show you the tail crest before it has been exposed to the gamma rays. And these are the plant variations of the tail crest after it has been exposed to the gamma rays. And you can see how it has like differences between the leaf size as well as leaf shape and the overall size of the plant. I will also show you a chronology about the development in crops using mutagenesis. I will give you a second to look at it. I will also show you a chart showing the diversity of mutant varieties that have been developed using induced mutagenesis with gamma rays. I invite you to do more research about nuclear chemistry as well as induced mutagenesis, and I hope you learned something today. Thank you for watching.